making RNA or DNA. They just, are useful to the cell as is. But of course, these are also the building blocks of RNA and DNA. These are the activated forms of the nucleotides. They will get added to a growing chain and lose a couple phosphates in the process. And finally, we probably won't get to it much this quarter, but there is another role for a couple of them that do this. So here's an adenosine monophosphate with the 5' carbon carrying the phosphate, but look what's happened here. The phosphate on the 5' has reached around and added to the 3' position too, making this a 3', 5' cyclic form of ATP. ATP and its cousin GTP both form cyclic nucleotides. These are generated when hormones arrive and bind to their receptors on the outside of cells. These become the so-called second messenger. The docking of the hormone on the outside of the cell triggers the formation of this inside the cell, in the cytoplasm. And this is what actually activates the hormonally activated processes. So there's energy uses of these nucleoside triphosphates. There's regulatory uses of hormonal regulation. And of course, they are the building blocks for nucleic acids. So here's the nucleic acid. Which one do we have here? RNA or DNA? Because? Yeah, no hydroxyl there. So notice we haven't drawn in the chemical structures here of the bases, just indicating ATG and so on. Because the focus in this slide is on the connectivity. How do you connect one base, one nucleotide to the next? You take the phosphate on the 5' carbon and you link it to the 3' carbon of a neighbor. So you've got these 5', 3' connections with phosphodiester linkages.
And another weird question.